Scopes, and we're just going to get started about the structures found within cells and how their structures relate to their function. Okay. So we talked about how the cell is like a factory. There are tons of things within cells, organelles, that make up a cell. And all these different things within cells, organelles, have a function. We're going to go over the names and functions of these different structures. We left off on slide 11. We left off on slide 11. I was showing you that cells can come in different shapes and sizes, but ultimately all of our cells have the same basic things inside of them. So I know when you look at a standard biology textbook, they show you a cell looking like this. Not every cell looks like this. This is just a generic cell. Some can be long, like muscle fibers, or very branched, like neurons. But they all at least have these basic components. So that's why we're just giving the standard looking blob of a cell and talk about what's inside of them. Okay, so let's start with if you need a packet, raise your hand. So we've gone over the fluid inside of cells. What's another name for the fluid inside of cells? Cytosol, yeah. So the fluid is cytosol or intracellular fluid. Remember we went over different fluid compartments? Intracellular fluid means fluid inside of the cell, intracellular. Cytosol, same thing. It's the solution of the cell. A solution is a mixture of water and other stuff. Like if you put salt into water, that's a salt solution, saline solution. If you put sugar in water, that's a sugar solution. There's a ton of different things mixed into the fluid within cells, not just those things. Amy, are there more? No. No more. Sorry. It's from last time, chapter 2. So, that in this fluid, the cytosol, you can find obviously water. That's what most cells. That's what the majority of cells are made of, water. You can find nutrients of different kinds. You can find waste molecules of different kinds. Something has to be encasing all of this, like a bag holding fluid. And so what's on the outside here, that's known as the plasma membrane. When you hear the word plasma, what does that make you think of? I know we went over plasma as a fluid in the blood, the liquidy part of the blood. It's separate. There's that word again, plasma. What does plasma entail? The word. What does it make you think of? Sticky? Or I kind of gave it away. Plasma is another word for what of the blood?
It's
It's a fluid. So this plasma membrane, it's not fluid exactly, but it's very fluid-like. It's very mobile. If you were to touch this at a molecular level, it would be able to shift really easily. It's because it's made up of a special lipid molecule called phospholipids, which we'll talk about more in a second here. It's made of a lipid molecule called phospholipids. The main goal of the plasma membrane is to protect. It's protecting all the stuff on the inside of the cell. And when you protect something, like if you have a city and you want to protect it, you build a wall around the city. You don't want the wall to just prevent everything from coming in. You want to regulate the passage of things. You want good things to come in. You want to prevent bad things from coming The term for allowing transport of some things back and forth, the word for that is semi-permeable. The word permeate means to go through, so only some things can go through. It's semi-permeable. You can turn your phone sounds off. You can see the arrangement of these phospholipid molecules. A single phospholipid molecule has a head region and a tail region. So here's a phospholipid. Here's the head. Here's the tail. So that's a single molecule. This is the first molecule we're going over, I guess, besides water. This is a phospholipid. And the way these phospholipids arrange themselves, they form two layers, and the tails face each other. So if you have a bag of phospholipids and mix it with water, they will always form, well, not always, but one arrangement that they can form naturally is this. We'll talk about why in a second. So like I said, the major component of the plasma membrane are these phospholipids, and they form a bilayer. Bilayer just means two layers. So there's one layer of phospholipids over here, and another layer of phospholipids over here. Again, one layer of phospholipids, another layer of phospholipids. It's the phospholipids that allow the membrane to be semi-permeable. I said that phospholipids are a type of lipid. Can anyone name another type of lipid that we use in everyday life? Sugar is not a lipid, it's a carbohydrate. Proteins, you both named two different classes. So there's lipids, there's proteins, there's carbohydrates, those are completely different. Give me an example of fat. Butter. Butter. Milk has fat. Yeah. Oil. Oil, great. Okay, let's go with oil, just because it's a simple example. When you mix oil and water, what happens? It separates. Okay, great. That's what I'm looking for. When you mix oil and water, it separates. This tail portion of the phospholipid, think of this as the most oily part of the molecule. It does not like water. Because these tails don't like water, they'll face each other. That's why they face each other, because they don't like water, they'd rather face each other. These heads like water, though. So they're facing that watery interstitial fluid and the watery intracellular fluid. Right, and because this is the oily part of it, it prevents things from easily passing back and forth. Molecules that don't mix well in fats or oils can't easily go through. So that allows it to be a barrier because of its chemical properties. That's as much chemistry as I want you to know at that level right there. All right, so that's a major component. It's a phospholipid bilayer. But it's not just phospholipids. There are other things embedded, like this weird squiggly thing, this blob here. There are other things embedded in the membrane. Two other things that I'd like you to know are cholesterol and proteins. Cholesterol, we hear this in the news as something that's bad and we want to limit it. We, like anything, we don't want anything, we don't need molecules in high amounts. Cholesterol in high amounts is bad for us, but it is an essential molecule. Cholesterol, one thing that it's really important for, represented here and here, these are cholesterol molecules and they help reinforce the membrane. They help add some reinforcement to it, some rigidity to it. Think of a plasma membrane as like the surface of an ocean. It's wavy. It's very fluid-like. What happens when waves get too big in an ocean? They crash and break. So in other words, the surface is breaking if the waves get too big. Something similar could happen to this phospholipid bilayer. It's wavy, but if it waves too much, it could break. And that would be really bad for the cell. The cholesterol helps prevent that. It's kind of holding it together, reinforcing it, preventing the membrane from breaking too easily. So that's the function of cholesterol. Yes?
Too much cholesterol, not let it move.
Would too much cholesterol not let it move? Yes, it would let it move less. But cholesterol, when you hear that cholesterol is bad for us, it's bad for you. It gets into our blood vessels and problems. Okay, we'll get to that later. But it's bad for a different reason. But if you have an excessive amount of cholesterol in the membrane, it would be more and more rigid. It would move less. Okay. Third thing that you'll find, third molecule type that we're going over, is a protein. I know you've heard this term before, protein. You've probably heard it as something that you find in meat and other high protein vegetables like beans. There's a lot of different things that proteins can do. So represented here in this picture, this is representing a protein, this is representing a protein, these are representing proteins. They can do different things. Proteins are really diverse and they have many different functions. So let's go over some of these functions. Here's one example of a protein and its function that you find in a membrane. If this is an illustration of a cell, here's the plasma membrane. These are proteins right here on the surface of the cell, embedded in the membrane. These molecules, these molecules that are binding here, this is insulin. Has anyone heard of insulin? What's that molecule for? It regulates your sugar levels. So let's say you just ate a meal, you've got sugar running around in your blood because you absorb sugar from whatever you ate. The whole purpose of eating sugar is to then bring sugar into your cells so your cells can use it. But we just said that the membrane prevents anything from coming into the cell. So we need a way to tell our cells it's okay to let things in. So insulin binds to this protein molecule. It's called a receptor. So that's what's shown here. Insulin binds to this protein, protein receptor. And then this receptor, when it gets activated, it tells the cell to do something. In this case, it tells the cell to let glucose into the cell. So here's now glucose being allowed into the cell. So that's our first example of a protein.
How do the glucose channels know to go to the edge?
How do the glucose channels know to go to the edge? It's complicated. We'll go over a brief example of how that happens. So sit tight on that question. What did I say? Receptors. So proteins can act as receptors. They can bind to molecules and then tell the cell what to do in a process that you'll learn about in physiology. You see, once this receptor is activated, we get other proteins inserted into here. These proteins are called transport proteins because they allow things to come in or out. So that's our second type of protein, a transport protein. Transport proteins span the entire membrane, and think of them as special passageways. They are special passageways. Like, let's say you're trying to get in someplace, but they won't let you in. So you just find like a back door to get in. This is that special back door. Transport proteins allow things that can't normally get through, like non-penetrating solutes, get from one side of the membrane to another. For example, glucose. It's a form of sugar. Does sugar mix well in water? Yeah, sugar can mix in water. If sugar can mix in water, does it mix well with oils? No. So in other words, glucose can't get through this middle layer because this is the oily part. So glucose can't just make its way through the membrane. It needs a special passage that'll let it through. It can go through a special transport protein made for it. So things like calcium, sodium, potassium, glucose, These need special ways to get in or out of the cell. Hopefully you're seeing that the membrane so far is pretty dynamic. It's very movable. You've got a ton of things embedded in it. And we will see that you can change what can go in the membrane. You can add stuff to it. You can remove stuff from it. Very changeable. Any other questions?
So sodium needs a transport protein, and then salt, like water is dissolvent.
Say one more time.
So sodium, that's salt, right?
Yeah.
It needs one of the transport proteins, even though salt would seem to dissolve in water.
Yeah, so salt dissolves in water, but does it still need a transport protein? It does. Anything that can dissolve easily in water generally needs a transport protein. There's always some exceptions, but anything that dissolves in water generally needs a transport protein. Unless it's really, really small. Yeah.
Does it never like just stay closed, like to something that it doesn't want to let in?
Does it ever stay closed to something that doesn't want to let in? It can, yeah. So in this previous example, this only opens up when glucose binds to it. It won't let other things in. I mean, water can get through because it can just slip right through. But most of these receptor or transport proteins are specific to certain molecules. So you have a specific transporter for glucose. You have a specific transporter for sodium. A specific transporter for calcium.
So do you like if you soak in sea salt water, would your cells know to open up and accept?
If you soak your body, any part of your body in salt water, would your body know what to do about it? Yes and no. There's another thing called tonicity, and that's getting into a different thing, which will we talk about? I don't know if we'll talk about it. We'll talk about that later, but that gets more of an issue. It'll try to take in as much as it needs. All right. Take a moment. Oh, sorry. Let me go back real quick. So we've just gone over 3 different molecules: phospholipids, cholesterol, and protein. Cholesterol is a form of lipid. Cholesterol is another type of lipid. So cholesterol is a lipid, phospholipids are lipids. Proteins are a different class of molecules. Proteins are a different class of molecules. All right, given that, here's a question for you. Raise your hand a few more times. Carbohydrates, proteins, lipids, and nucleic acids. Those are biological. Raise your hand a few more times. Hands on 3. 1, 2. Talk to your neighbors real quick. Go ahead. All right. Let's see your answers one more time, please. Hands on three. 1, 2, 3. On 3, the letter. 1, 2, 3. Yeah. Membrane transporters and receptors. What kind of molecule makes those? Proteins. We haven't gone over D yet, but proteins also make that up. Like cells can be held together. Proteins do that. Major component of membranes is always phospholipids. So we've talked about the plasma membrane, what's on the outside of a cell, and about other membranes that are on the inside of the cell, which are also made of phospholipids. Here's another question. If you need help visualizing this, here are cells of the digestive tract. And then, yeah, here's where food's passing through, and here are cells lining it.
It's not interesting.
Raise your hand a few more times. 10 more seconds. Okay, let's see what you think. Hands on 3. 1, 2, 3. Take a moment, ask your neighbors. 1 minute. Find someone with a different answer. Find someone with a different answer so you can argue on right. Why?
Both breast and egg. But
Oh, I always have five.
if it's not gassy in between cells, yeah, it's great to be
Okay, so you both agree. You have a different answer? Okay. Do you agree? Wow. It says it can't pass between. Prevents multiple. All right, one more time, please. Hands up. 1, 2, 3. All right. So here we have, let's say this is your digestive tract. What's the term for the space where food is passing? That's the lumen. Okay, you can agree with that. Oh, they all say that. Okay. So we got nutrient molecules here in the lumen. We need to get this through these cells. There are these special proteins called tight junctions that prevent these molecules from passing in between the cells. So they can't go in between the cells. That can't happen. Instead, they have to go through the cell. Any guesses what would let it into the cell? What on this cell would allow it into the cell? Not a receptor, a transport protein. Transport protein. So there's going to be transport proteins on this cell that allow the molecules to go through. So it's going from the lumen to the intracellular. And then it can go to the interstitial, passing through the cell. Any clarifications on that? So the answer is A. The benefit of this, as I mentioned, why bother having different fluid compartments? The benefit of this is now this cell can screen for these molecules that are passing through. When we eat things, we eat nutrients, of course, but sometimes we eat things that are harmful to us, whether it's toxins or bacteria. This, going through a cell here first, gives it a layer of screening. It's like going through TSA. The answer is A. Questions? Okay. Okay. So we've gone over the cytosol, we've gone over the plasma membrane. Now we're going over that major organelle on the inside that you all saw the other day, the nucleus. The word nucleus just means the center, the core of something. So the core of the cell, shown there in that magenta color, that's the nucleus. The nucleus is an organelle. It's an organelle made of membrane. So this is zoomed into the nucleus. So here's the membranous part of the nucleus. What's this membrane made of primarily? What's this membrane? DNA. It stores DNA. Inside of it is DNA. What's the membrane made of? What's the plasma membrane made of? What's this membrane made of? Phospholipids. Okay. Every membrane, if you ever use the term membrane when we're talking about cells, those membranes are made of phospholipids. If we're talking about the peritoneal membrane, that's different. At a cellular level, when we're talking about membranes, they're always primarily made of phospholipids. So this membrane is primarily made of phospholipids. Okay. What's in it? That's DNA. So inside of it is mostly DNA. All the genetic information that tells our cells what to do. So that's the general function of the nucleus. It's a protective area to store DNA. Not only does it protect it, it's not just a safe house for it, but it also regulates its usage. Just like if you have something in a safe, but let's say you need to go in and out of it all the time, you want to regulate when that comes out and how it's protected. But the DNA always stays in the nucleus. It should always stay in the nucleus. DNA is so valuable, so we always need to keep it there. Let's say you have a really expensive book in the library, really rare, and you don't want to damage it, but you want to read it. You're not allowed to take the book out of the library, so what do you do with the book? I mean, you can go in and read it, but let's, you don't want to steal it either, that's bad. You want to read it at home, but you can't bring the book out of the library, so what do you do? Take pictures, I guess that's the new thing. Take pictures or copy it. You want to copy it. You want to scan it. So the DNA has the instructions, but we can't take the instructions out of the nucleus. So how do we get the instructions out? We scan it. The term for that scan thing is called RNA. RNA is the molecule. It's basically a scan of the DNA, but you can take this out of the nucleus. The DNA stays in the nucleus protected. This is a copy, well, not quite a copy, but more of a transcript. This is a transcript. If you damage it, it's not the end of the world. If you damage your DNA, that can lead to things like cancer and mutations, and bad mutations and other things like that. But this we can take out, and it can be destroyed, and it's no big deal. These instructions come out of the nucleus, and they make their way to another organelle. These organelles are called ribosomes. Ribosomes. Ribosomes will read these instructions. The major reason why we have DNA, there's a few other reasons. The major reason why we have DNA is that there are instructions on how to make protein. DNA are instructions on how to make protein.
So do the ribosomes read the RNA or the DNA?
So the ribosomes, what will they read? They will read the RNA because the DNA stays in the nucleus. Ribosomes are outside of the nucleus. So all these little specks here, those are ribosomes. Those are representing ribosomes. Here in this micrograph, you can see these specks. Those are ribosomes. What kind of a microscope was used to make this? Transmission. We're looking inside of the cell. You see those little spots.
We want to make more cells. Do we take it from the RNA? Or do we...
When we want to make more cells, where do we? We'll get to that. When we talk about mitosis, let's make more cells. How about that? Right. The whole goal of DNA has instructions to make proteins. But first, we have to make RNA, and then RNA makes its way to the ribosomes, and ribosomes will manufacture proteins. So think of ribosomes as your workbench in the factory. This is where they're your assembly workers. They're putting things together based on instructions. Where's the RNA located? RNA is located, it starts in the nucleus, this is where it's made, and then it comes out of the nucleus and makes its way to ribosomes. So the ribosomes they might be floating around the cytosol, but they're generally attached to this other membranous organelle called the rough endoplasmic reticulum. Really fun long name here. The word reticular, that's Latin for a network. So it's a network of membranes that's inside of the cell, inside of the plasma membrane. And that's what endoplasmic is referring to. It's a network of membranes inside of the cell, and it looks rough. When you look at it under a microscope, it looks rough. It looks rough because the ribosomes are attached to it. It gives it that texture. That's why it's called the rough endoplasmic reticulum. Let's say you're in a factory and the first thing is to assemble all the pieces together. Let's say we're a box factory, we're making boxes. So you start with a really huge piece of cardboard, but then you have to cut it to the right shape. And then put the right creases in the box that will eventually get it to a form of box. The ribosomes are the ones cutting and shaping the cardboard. Once we have our rough product, it gets inside of the rough endoplasmic reticulum. The rough ER, it doesn't create the protein, but it folds the protein. It makes it functional. Like if you have a box that's flattened out, it's not functional. For a box to be functional, you need to fold it and tape it into the right shape. That's what happens in the rough ER. It processes it, it folds it into the right shape. So the rough endoplasmic reticulum its purpose is to process, to fold protein into its correct shape, into its correct three-dimensional shape. Yes. Yeah, where is this found? It's kind of all over the cell, but generally around the nucleus. It's outside the nucleus, within the plasma membrane. Where are ribosomes? Ribosomes are found on the membrane of the endoplasmic reticulum. What's the membrane of the endoplasmic reticulum made of? Phospholipid. All membrane. Okay, so again, our cell is a factory. We have instructions on how to make protein. That's DNA and RNA. We use those instructions to make protein. That's what ribosomes do. Once the protein's made, we need to fold it properly. That's what rough ER does, rough endoplasmic reticulum. Once you've made your product, if you're a factory, you want to do your product. Ship it, sell it and ship it. That's, here's an example of my box folder. And, then, and there's an animation of a protein folding. Oh, there it goes. So that all happens in the rough ER, the proper folding of it. Okay. Next in line is this organelle called the Golgi complex. Golgi is a dude's name, scientist who discovered a bunch of things, including this organelle. The Golgi complex is another membranous organelle that takes the protein from the rough ER and then sorts it. Think of this as your shipping center. It sorts it and then ships it to the correct destination. We've gone from the nucleus. We had information inside of the nucleus. We brought that RNA, so this yellow strand is the RNA. We brought that RNA to a ribosome. These ribosomes are the purple ones. The ribosome produced the protein. This protein is within the rough endoplasmic reticulum where it gets properly processed and folded. And so here's our complete folded protein. We send that complete folded protein to the Golgi. Here's the Golgi complex. And the Golgi will sort it and ship it to the correct place. What is the correct place? Proteins are used for a million different reasons. We could send it somewhere else in the cell, because our cells need proteins. We could send it to the membrane, because as we've seen, membranes need proteins. Or it could be sent out of the cell, because sometimes we need to send proteins to other places. So that's what the Golgi is deciding. It's figuring out where should this protein go. How does it send all these proteins around? I said that all these things are made of membranes, and the membranes are very fluid. Phospholipids are very fluid. So think of them as like if you're playing with Play-Doh, you can pinch off a piece of Play-Doh, but then you can also add that piece of Play-Doh to something else and it'll just mix in like it's nothing. You can pinch off membrane from the rough ER and have it integrate itself into the Golgi. You can pinch off a piece from the Golgi and have it integrate itself in the plasma membrane because they're all made of the same things. The term for this pinched off membrane, that's called a vesicle. The term for a pinched off membrane is called a vesicle. And that's how proteins can be transported around within these vesicles. So for this protein to get from the rough ER to the Golgi, it uses a vesicle. For this protein to get from the Golgi to the plasma membrane, it uses a vesicle. Golgi complex, Golgi apparatus, Golgi body, they all mean the same thing, yes. You can, I might even just call it the Golgi because it's easier. But Golgi complex, body, apparatus, they all mean the same thing.
The vesicles go outside the cell.
Do the vesicles go outside of the cell? Good question. Say you have this vesicle, and you can see the proteins on the inside. So you can imagine this vesicle will then fuse with the plasma membrane. Will the vesicle leave the membrane?
No.
Well,
Open up.
it will open up, and what happens? What happens to this membrane?
They form the membrane outside.
Sorry?
Forms
It contributes. It becomes a part of the plasma membrane. So this membrane from the Golgi can become part of the plasma membrane when it fuses with it. It becomes a part of it. Then adds to it. Yeah.
And does it like re-separate back to like the Golgi complex back together?
Yeah. So the question is, if you keep pinching up vesicles, that means this is going to get smaller and smaller. The reverse process can also happen.
How long does this whole process take?
How long does this whole process take? That's a good question. I want to say it's at like seconds time scale, like seconds.
Okay, so if you were to look under like a microscope and be able to see this happen in real time, it would be pretty quick.
Yeah, if you could see this under a microscope and see this happen in real time, yes, it would happen pretty quick.
Can you put your hand under transdimensional microscope?
Can you put your hand under? No. Can you put cells? Yes. So we can like culture tissues or have a tissue sample. What do you do to live tissue now? I mean, you can culture living tissue. And try to do this. You can culture our cells or any other cells.
When you put a sheet cells under the microscope, how come you see it
How come we didn't see it?
like you mentioned?
Two good questions, or a good question here with two answers to it. When we removed our cheek cells and looked at it under the microscope, how come we didn't see anything happening with those cells? There are two answers to this. One,
You need a transmission electron.
you would need a transmission electron microscope to see anything at this level happening. But what's another reason? Anyone else? They're dead. These cells are dead. Yeah. The outer layer, we're going to learn that the outer layers of your skin and your mucous membranes are dead. Which makes sense because if they're going to fall off easily, you don't want them to. They're just like a protective scale. Cool. Oh, so here's an example of that fusion with a membrane. So here's a vesicle. It fuses with a membrane and then it can let stuff out.